All right, so good afternoon, everyone. Thanks very much for being here. We're very fortunate to be joined by Dr. Lawrence Lustig today, who will be speaking to us about genetics, the genetics of hearing loss. Dr. Lustig is the Howard W. Smith Professor of Otolaryngology, Head and Neck Surgery, and also Department Chairman at Columbia University Medical Center, where I did my residency. Dr. Lustig is a surgeon scientist and an international expert on the genetic mechanisms of hearing loss, auditory neuroscience, and utility of gene therapy for treating hearing loss. He received his MD from UCSF and completed his residency as well at UCSF and went on to complete a fellowship at Johns Hopkins in neurotology. He remained there on faculty at Hopkins for a number of years before moving to UCSF to become an endowed professor, where he was until 2014. And then he ultimately joined Columbia as department chairman thereafter, where he has been since. He's a prolific researcher and has had extensive experience working on delineating the genetic mechanisms of hearing loss and how gene therapy might be used to treat genetic hearing loss, both in the basic science as well as clinical research realms. And professionally, he served as the editor-in-chief of Otology and Neurotology, as well as being the past president of ARO and the American Neurotologic Society. So with all that, thank you so much for being here today with us, Dr. Lustig, and we look forward to your talk.
Fantastic. Thank you so much, Josh. I hope you are all enjoying Josh as much as we enjoyed him here in New York. He really is an amazing he was an amazing resident. I'm sure he is an equally amazing fellow. Now I got to figure out how to share this. Wait, where did it go? Unbelievably. Okay, this is crazy because now my PowerPoint is no longer coming up as an option. Hold on one second here. Wait, where did it go? Oh, cancel. My PowerPoint disappeared from my share tab. This is crazy. Let me log back in and see if I can find this again.
You know, we can also try it. We can try it this way. If you can you see your slides on my screen sharing now?
I can see yours, absolutely. Yeah.
So I can do that. You just have to tell me when to advance.
All right, let's advance then. Perfect. Okay. So okay, advance. Nope. No, that's not it. There we go. One more. Perfect. Okay. So thanks everybody. Sorry about the sort of the Band-Aid approach to this. Anyway, we're going to talk today about the genetics of hearing loss, which is really an amazing topic this day. It's so relevant in terms of everything that we're doing in terms of caring for patients and a whole spate of treatments that are just around the corner. So I'm going to start by talking a little bit about the history of the genetics of hearing loss, because I can't help myself. I love history. We'll talk about sort of the underlying molecular genetics of hearing loss. We'll talk about genetic testing. And lastly, we'll talk about gene therapy as a treatment for many of these conditions. Next slide, please. So as you can probably imagine, it's been known that hearing loss has been passed down from generation to generation. As early as the 1600s, Paul Zacchius even had this great quote about the deaf and the dumb ought to abstain from marriage for the good of the commonwealth, because they beget children like themselves. And as politically incorrect as this would be to say today, it really recognizes the fact that it's been known that if there's hearing loss in a family, it can go down through the generations. Next slide. And in fact, Adams described in 1840 4 generations within a single family with hearing loss, and he was also the first to recognize the genetic and hereditary basis of otosclerosis. Next slide. And interestingly, Martha's Vineyard, which is just off Cape Cod, was known for many, many years to have an inherent sign language called Martha's Vineyard Sign Language. And there, for a long time, many patients on Martha's Vineyard, all of whom descended from a single individual, had deafness. This was studied originally in the Lancet in 1877, but also there's a book written about this. So again, another example of just long-standing families and generations of patients with hearing loss from a single individual. Next slide. So William Wilde, interestingly, the father of the acerbic playwright Oscar Wilde, also recognized the importance of parental deafness and autosomal recessive hearing loss as early as 1873. This was denied and argued against by George Darwin, who interestingly was the uncle of Charles Darwin and would ultimately provide the evidence that in fact there is a genetic basis for hereditary disease. Next slide. So then if we go through all of the genetic steps that happened, the large genetic advances that happened since Darwin's great discovery, including De Vries, all the way up to Avery, who understood that there was a genetic component that was passed between bacteria. Beetle, the great work of Watson, Crick, and Francis. Gamow, who first understood that it was nucleic acids. Nuremberg, who was the first to solve the first genetic crack of triple U for alanine. Kary Mullis, who developed PCR. And fast forward all the way to the next slide, which was the Human Genome Project. This was begun in 6 countries in 1990, an estimated price tag of $3 billion over 15 years. And for one of the first times, it was done under budget and under time, finished in 2003. Really a remarkable achievement, which was the engine for really allowing us to understand a number of genetic diseases, but most importantly, genetic deafness. Fast forward, next slide. So you all are well versed in the ear anatomy, how sound comes down the ear canal, through the middle ear bones to the inner ear, where those sounds are converted to electricity, which goes to the brain. Next slide. Now, what we're going to do is we're going to focus in on the organ of Corti, which is really the engine of hearing. Next slide. And if we really focus even more closely on the organ of Corti, next slide, you can see the fine structure of the inner ear. So we've got the inner hair cells outlined in red. We have our 3 outer hair cells. We have our tectoral membrane and all of the connections between these things. Now, it's amazing how much of this has been elucidated by our understanding of the genetics of hearing loss. I'm going to give a couple examples, but next slide. So if you look at the fine structure of the hair cells, for example, we know they're hair cells because of these small stereocilia that project from their apex. Next slide. These stereocilia are loaded with not only ion channels, but also tip links that allow them to do their job. And as you likely know, as these hair cells deflect, these tip links open up and allow potassium to rush into the hair cells, propagating the neural signal to the central auditory pathway. And all of these proteins and ionic channels were developed from research trying to understand the underlying molecular pathways of hearing, and in turn informed us about how each of these, when mutated, can lead to hearing loss. Next slide. And this is just a high resolution example of these tip links that are so critical for hearing that were developed by some of these studies. Next slide. So moving on to hearing loss, as you know, about 1 in 1,000 children are born with congenital hearing loss, and about 60% of these are genetic. Of these, about up to a third to a half have an inherited component. Next slide. And of these, we divide these into both syndromic versus non-syndromic. Syndromic forms of hearing loss account for about 30% of cases of inherited hearing loss. And by definition, these are systemic gene functions. So for example, if you've got a gene that's mutated in multiple tissues, you'll get a variety of problems, including maybe blindness or skin color pigmentation problems. They're part of a recognized syndrome. In contrast, non-syndromic forms of hearing loss are only seen as effects within the inner ear. These count for a majority of forms of inherited deafness, about 70%. And we can divide these into 2 very broad categories: DFNA, which is autosomal dominant, and DFNB, which are autosomal recessive. There's a third category, which are mitochondrial, but we won't be talking about those today. Next slide. So our understanding of the genetics of deafness has really come from 3 major lines of research. Number 1, large consanguineous families with hearing loss, where you can do the traditional linkage type analysis studies to identify potential genetic markers. Number 2, genetic mouse models of individual genes that were mutated that allow us to see what the effects are in a biologic specimen. And then combining this with the information that has been gradually coming out of GenBank that allow you to then correlate what you see both in families and in animals. And this is an iterative process that's been going on over and over. And what I like to do is talk about just a couple examples to give you a sense of how all of the different forms of genetic deafness have been elucidated. Next slide. And I divided these into sort of functional properties. So the first ones I want to talk about are some structural proteins in the inner ear. Next slide. So one of the classic examples is Usher syndrome. Now, Usher syndrome is an autosomal recessive disease. It's the most common form of combined deafness and blindness. And it's an instance of about 4.4 per 100,000 individuals. And by definition, it's syndromic because you see manifestations outside of the ear. Next slide. The blindness is due to retinitis pigmentosa. And the blindness can come at any point between, childhood and adulthood. Next slide. And if you look at the inner ears of patients with Ushers, you see a complete absence of the organ of Corti and a near total loss of the spiral ganglion neurons late in life after they've donated their temporal bones to science. Next slide. So we divide Usher syndrome into three types. Type 1 is the most severe. Patients oftentimes born with profound hearing loss or develop it very early in childhood, typically within the first 10 years, oftentimes with absent vestibular function. Type 2 is a medium term. Patients will have a downsloping hearing loss with onslope within the first 20 years. Type 3 is the least severe. It's progressive with variable responses. And for a long time, we thought, are these three different types of diseases, or is this the same mutated protein in different ways? Next slide. Well, the first clue came from David Corey's lab. David Corey is a very well-known auditory researcher. He was studying this one particular protein called myosin. Myosin, as you know, are parts of muscle movement, but they're also involved in molecular transport. And David Corey was the first person to propose that there was the deflection-gated ion channel that allowed the inner hair cells to propagate the auditory signal, what we had just discussed a few minutes ago. And he proposed that these myosins in their ability to move allowed the channel to reset. So in a sense, once that hair cell deflected, it wasn't continuously sending a signal to the brain. It allowed it to reset so it can be ready for the next round of auditory signaling. Next slide. And again, myosins are critical not just for muscle contraction, but also for molecular transport. And this made a lot of sense. Next slide. And he proposed again that this stereotip link was like a spring-gated ion channel, and as that spring deflected open, after it allowed potassium to rush in, the myosin would allow this to crawl back up one of the stereocilia and reset closed again. Next slide. Well, at the same time that Corey proposed this, there was another group led by Gibson that was studying a well-known mouse called the Shaker-1 mouse. This mouse was deaf and had loss of vestibular function. And it was determined that the deafness in these mice in 95 was due to abnormal mutations in this myosin gene, the same one. And this was the first molecule that was known just by virtue of a mutation that was involved in auditory transduction. Next slide. So completing the circle, while in 1995, in that same year, was studying the human counterpart, which is the myosin 7A gene on chromosome 11. Now, this had previously been linked in the general region with Usher syndrome. And he was studying 9 unrelated Chinese families with Ushers. And in 2 patients, he was able to trace that there was a mutation in the myosin 7A gene, finally creating that feedback loop that allowed him to determine that myosin was not only the causative problem in Usher syndrome in those patients, but also was due to an abnormal myosin and actually how it caused the deafness in those families. Next slide. Well, to make matters even more complicated, that same year, Christine Petit at the Pasteur Institute was studying a Tunisian family with autosomal recessive deafness, non-syndromic deafness. And in fact, she showed that myosin 7A, when mutated in a different way, caused non-syndromic deafness, which has been termed DFNB2. Next slide. So in fact, this is something that we have seen over and over in the genetics of deafness. You take one protein one gene, you mutate it one way, and you may have a premature truncation and complete absence of auditory function, and hence you end up with a syndromic form of hearing loss, very severe. In contrast, you may have an alternate mutation, and in this case, decreased splicing efficiency by 40%, causing reduced function of the protein, but still presence of the protein, and here you get non-syndromic hearing loss. And in fact, we've seen this exact same thing for a number of other syndromic and non-syndromic genes. Next slide, please. So another example, and now we're going to go down to Costa Rica, and this just demonstrates the importance of large kindred families for allowing us to understand the genetics of deafness. Next slide. In this case, Leon and colleagues, there was a large 147 member of the Costa Rican family, 78 of whom had hearing loss. And what they did, they just did hardcore genetic linkage analysis. And this was back in the 1990s when we didn't have access to next-gen sequencing, and you had to do classic Sanger sequencing. It was like looking for a needle in a haystack. And they did this, and next slide, please. And what they found was that a suspected portion of chromosome 5 where they linked the deafness, they just did the hardcore sequencing. They found this gene called diaphanous. They went back to GenBank and showed that, in fact, this was the gene that was consistently mutated in these families. Then they just went into the inner ear, just looked at the mRNA and showed that diaphanous was, in fact, produced in the human inner ear, linking diaphanous to the deafness in these families. Next slide, please. So it turns out that this protein is in the reticular lamina at the very top of the hair cell, right where the stereocilia insert into the hair cell. So if you have loss of that function of that firm hold of the stereocilia, it's another form of deafness. Next slide. These patients with DFNA1, it was the very first autosomal dominant form of deafness described, have a low frequency hearing loss. This works with two proteins called Rho and profilin, and these promote actin polymerization. So again, it turns out that the hair cells are exquisitely sensitive to actin polymerization and a number of other proteins that, when mutated, that are also involved in actin function, also lead to genetic deafness. Next slide. Well, as you can guess, because the inner ear is one big battery and we need that charge of battery in order to hear, it shouldn't be surprising that ion channels are also critically important in hearing, and mutating these proteins will lead to deafness. Next slide. And if you just look at the sheer number of ion channels in hair cells, they are just chock full of them, as you can see in this illustration here of some of those proteins. Next slide. Well, it turns out one of the most important are connexin 26 proteins. These are gap junctions. These are non-active proteins. They're just simple benign electrical channels between adjacent cells that allow the free transport of potassium, chloride, or sodium. Next slide. Well, Kelsall was studying a large family with skin problems and deafness, and he was in a dermatology department searching for the skin related issues, but it turns out he identified connexin 26 as causing the deafness. And it turns out that connexin 26 was the culprit in a number of families of inherited non-syndromal deafness that had previously been described, including two forms, DFNB1 and DFNB3. Next slide. So it turns out that potassium transport is critical in hearing. As you know, we have a potassium recirculatory pathway. The stria vascularis pumps in potassium into the scala media. That then gets deflected into the hair cells when you hear. But that potassium has to be cleared out of the hair cells. Otherwise, that increased potassium is going to lead to hair cell death. So there's a whole clearance pathway through the supporting cell network back up to the stria vascularis, which then pumps it back in to the scala media. So it's a circulatory pathway. Well, connexin 26 is critical in this. And if you block out connexin, that potassium builds up and leads to a potassium-induced cell death. Next slide. So this is caused by the gene GJB2. And in fact, it's the most common cause of non-syndromic deafness. Depending on which study you're looking at, anywhere between a third and a half of cases described. Most of these are a single type of mutation, the so-called 35del G, which gives you a frame shift and truncation. These account for about a quarter cases of sporadic deafness. And a study we did a number of years ago in our CI population, about 1 in 6 of our patients had some form of mutation in connexin 26. Next slide. Another ion protein, which is critical and very important to identify early on, is KVLQT1, which works with another protein called KCNQ1. This is responsible for Jervell and Lang-Nielsen syndrome. This is what causes prolonged QT syndrome and sudden death in children and adolescents. So this is something that's pretty critical to identify early on. And that is one reason why, as part of the genetic hearing loss workup, we always get an EKG, because if they have this protein problem, you want to be able to identify it early. This is also responsible for another type of deafness called Beckwith-Wiedemann syndrome. This is a germline chromosomal rearrangement, and patients get prenatal overgrowth and predisposition to childhood cancers. Next slide. Well, this is a voltage-gated potassium channel, also critical for hearing within the stria vascularis. And if we don't have potassium circulation, as you know, the patients will go deaf. Next slide. And this is an example of a knockout mouse that we studied many years ago. We knocked out KVLQT1, and it turns out the mice were, in fact, absolutely deaf, as you see from the AVRs here. Next slide. So the knockout in mice, the stria vascularis is where the KVLQT1 is localized, as you'd expect. Deafness in the knockout mouse. And again, another example of how potassium recirculation is absolutely critical to hearing in humans. Next up. Now we go down the list of all the syndromic forms of hearing loss, Alport's, BOR. We're not going to go through every single one of these, but you can see, these are for various structural proteins like collagen, more for the stria vascularis, potassium channels, mucin-like proteins, PDS sulfate transporter genes, structural genes, etc. Next slide. And if we look at all the locations of some of these proteins in the inner ear, this is from the Hereditary Hearing Loss homepage. Next slide. You can see that we've got a number of genes that are localized just in both outer and inner hair cells, all of which, when mutated, will lead to deafness. Next slide. We also have some genes that are specifically just localized in the outer hair cells. Next slide. As well as just other genes that are localized just only in outer hair cells. Next slide. And cells that are just in supporting cells. Next slide. Spiral ligament proteins. Next slide. Tectural membranes. Next slide. And proteins isolated in the stria vascularis. Next slide. And you can also have a number of different genes when mutated that can lead to deafness that are found throughout the inner ear, including the GJB2, connexin 26, other forms of connexins, and a number of other genes as well. Next slide. And we can even focus in specifically on the stereocilia themselves within the hair. So if you look at all of the different genes on this left-hand panel, each one of these, is, when mutated, will lead to deafness, and have all been described in humans. Next slide. So to date, we have over 100 signal deafness genes identified at 140 different loci, and it's predicted that we're going to finally end up, by the time all the dust is settled, over 280. It's really fascinating, because every other group that studies the genetics of disease, when they look at hearing loss, they roll their eyes. They can't believe the complexity of this problem. So it's really unique among all of the different genetic diseases in the world that are studied. Next slide. So, I outlined some old examples where we had to go through classic linkage analysis studies and old classic Sanger sequencing. But we've really entered a new era, what's called next-gen or next-generation sequencing. And if you look here on the left-hand side of the cost of the gigabytes per database, if we look originally at the original Human Genome Project of $100 million, all the way through 2016, you can see that the cost has gone down exponentially. Meanwhile, the gigabytes output per week has also risen exponentially since the original sequencing was done. This is a slide from Illumina. I don't make any money from them. I have no conflicts with them. Next slide. So next-gen sequencing really has revolutionized how we look at genetic disease. So if you don't know how it's done, basically you've got a reference genome sequence. You take the genetic sequence that you're looking for, and then you chop it up into millions of different pieces. And each piece then has tags on either side that allow you to sequence that individual sequence. Then using massive bioinformatic algorithms, you put it all back together as a puzzle, and then you can then reference, you can then compare that to the reference sequence and determine whether or not there are single nucleotide polymorphisms or SNPs, or there are any mutations, or if there's any deletions or even duplications. Next slide. And what this allows you now to do is then compare large individuals. So you can have a group of patients with disease, compare these with a large group of controls, and you can do whole exome sequencing, which is basically all of the active bits of messenger RNA being sequenced. Whole genome sequencing, you can do targeted sequencing, just like, like gene chips. You can look at single SNPs, and you can do all of these different analyses. And then from that, you can come up with an alleles associated with the disease odds and an odds ratio. So you can determine whether or not that particular abnormality you've identified, what's the prevalence in the disease group versus the control group. Next slide. The first example this was done, which was really quite shocking, was described in 2005 by Klein. They were looking at age-related macular degeneration, and completely by shock, they found abnormalities in complement factor H, something that you never would have identified looking at individuals or even single families. And that's the power of next-gen sequencing using large populations of individuals. Next slide. The first example in our field was done by Rick Friedman. He's now at UC San Diego. He was working with a European group, and they had a large group of patients with age-related hearing impairment. And they compared that to a reference gene set, and they identified 3,400 individuals. And they did a single nucleotide polymorphism analysis, SNP analysis, and identified 2 SNPs that were located on a single gene, which is called metabotropic glutamate receptor 7, or mGluR7. And when you looked at the inner ear of these patients, it was localized to the spiral ganglion neurons. So definitely a relevant hit. Unfortunately, this has never been replicated in any other group, but nonetheless, it was an important study to demonstrate the power of doing whole genome association studies, or GWAS studies, for hearing loss. Next slide. There's been a number of reports since that seminal report. This is one study. They looked at whole exome sequencing on 100 patients. And this is really interesting. They found 5 variations in different syndromic hearing loss genes. None of these patients had syndromic hearing loss, but all of them had abnormalities in the syndromic hearing loss genes. This is identical to the Usher story that I told you originally, where the gene mutated one way gave you Usher syndrome, but a different way gave you a non-syndromic form of deafness. It's replicated here on 5 different examples. Next slide. There's another, we did a GWAS study in the Kaiser Permanente database back in 2017. We looked at 6,500 cases of age-related hearing loss compared to 45,000 controls, whole exome sequencing, and we found 5 different potential genetic causes of hearing loss. The main problem with these large databases is that you don't necessarily have really well characterized disease populations. It may be you may get patients check off they have hearing loss on a health screen, but that could be hearing loss from conductive hearing loss, that could be a genetic hearing loss, that could be from a gunshot noise, right? So this is where these studies can get a little bit messy. Next slide. And these studies just add up. And actually Jasper in 16 showed that over the prior 5 years, next-gen sequencing was responsible for discovering 1/4 of all of the genes known to cause non-syndromic hearing loss. And I think over the next sort of decade, this is where we're going to find all the remainder of the non-syndromic hearing loss genes. Next slide. So here we are truly entering the era of personalized genetic medicine. Next slide. So one of the issues that we're having is sort of I think we're still in the Wild West of genetic testing. In 2016, when this study was published, there were 5 genetic laboratories. Each of them were targeting a substantially different number of genes, anywhere between 27 and 150. So, you may have a patient that went to one of these labs that only looked at 27 genes, and they will come back and tell you, hey, I came back negative. But in fact, if they had a different test, you may have identified what the genetic mechanism was. Currently, most groups are starting with a staged approach. And using the best available analyses, about 2/3 of patients should be able to come up with a genetic cause of hearing loss if you strongly suspect a genetic basis. Next slide. And it's important, if you're going to start doing genetics testing, to know where you're sending your genetics testing to. How is that lab validating their genetic variations they've analyzed? Which genes are they analyzed? What's their detection? Are they looking not just at genes, but at copy number? Are they looking at deletions and duplications? And what's the cost and turnaround time? Because it's widely variable even today. Next slide. And this is just an example of some of those differences, right? Here's costs range anywhere between $596 to $3,200, with a huge numbers of different genes that are being analyzed. So there's no standards for this. Even today, there's no standards. So if you are sending your genes out, know the lab where you're sending her out and know what tests you're getting. Next slide. And this came out from the International Journal of Pediatrics. This is the International Pediatric Otolaryngology Group consensus recommendations. At one point, about 10 years ago, it was recommended to start with GJB2 analysis, and that's all you needed to do. But now that is not recommended anymore. You're going to miss a whole lot of genes, even though that's the most common. Its current recommendations are comprehensive genetic testing, either whole exome or whole genome analysis, and also a stepwise approach. So it's perfectly acceptable to start with the known genes that we have, like on a chip. And if that doesn't work, then go to whole exome. And if you still don't find anything, then go to whole genome. That's the paradigm that will get you about 2/3 of a hit rate with current genetic testing models. Next slide. So in spite of all we know about testing, we still are pretty rudimentary in terms of how we're treating patients. Next slide. For mild to moderate hearing losses, even some form of severe hearing losses, we're still offering hearing aids to everybody. Next step. For patients with severe to profound hearing losses, as you know, we give them cochlear implants, and they work great. There's no question, but I don't think anybody could argue that natural hearing would be better than what you would get out of a cochlear implant. Next slide. And this is where the promise of gene therapy is really sort of coming through. Next slide. So when we talk about gene therapy, we're talking about how to modulate the underlying genetic base, the underlying genetics of individual cells. So we have a couple ways that we can do this. We can try to modulate a signaling pathway, and the classic pathway within the inner ear is the ATO1 notch pathway. And this has been widely used to try to regrow hair cells. This was used in a Novartis study, genetic therapy study that we did a couple of years ago. That was a bit of a bust. It's currently the paradigm that's being used for the frequency study that's being done out of the company in Boston to try to regrow hair cells. But for genetic forms of deafness, we've got a couple options. We can try to restore absent function. We can try to override a defective protein, or in the case of a heterozygous dominant mutation, we can try to inactivate the defective gene. Next slide. And there have been a number of studies using all three of these different methodologies, from VGLU3 in 2012, which we described, all the way through Connection 26, several forms of Usher syndrome, a number of different forms of non-syndromal deafness as well, up to most recently otoferlin. Next slide. Now, unless you've been living under a rock, you've probably heard about CRISPR-Cas9. This is sort of a gene editing methodology. You can actually go in and rewrite the genetic code. The advantage of this is you can take care of this problem not only in the person that you're trying to treat, but also potentially in their offspring. There are some issues with CRISPR-Cas9 right now. It's hard to get high enough levels in the target protein, but in spite of that, this group out of Harvard, Xu Gao's lab, they were studying in the Beethoven mouse which is the cause of several different forms of non-syndromal deafness. And this is a heterozygous autosomal dominant mutation, meaning you only need one bad copy to cause the hearing loss. So what they did with the CRISPR-Cas9 is they targeted the mutant allele to cut it out of the genetic code. And by doing that, they reduced the progression of the hearing loss. They didn't actually cure the hearing loss itself. So it was a bit of a win, but nonetheless, it was an innovative way. And I think this may be a way to sort of target heterozygous dominant mutations in the future. Next slide. And this is a recent study that we did in mice, where one of the main limitations we have with gene therapy is the sheer size of the gene that we can put in the available vectors that we can get into cells. And AAV, or adeno-associated virus, has a pretty severe limit in the size of the DNA that you can put in it. But this is the way that we actually took two different vectors, split up the gene into two different parts, put on some sticky ends so that when they both recombined within a cell, if they both were transfected, you get a full-length messenger RNA. And we did this for otoferlin in a mouse model of deafness, and we're able to completely cure the deafness in this mouse model. And this also greatly expands the number of genes that you can use for gene therapy using AAV, which is our preferred mode for hearing. Next slide. So curing genetic deafness using gene therapy is not just simply getting the gene in the right place, but there's a whole process here, right? Number one, we need to have universal newborn hearing screening, which is critical. We do that right now, so I've given that a green. But we also need to actually start having universal newborn genetic testing, because a lot of kids are going to be born with normal hearing. But you need to know if they're going to develop hearing loss early on so you can intervene early, because we know now in mouse models, we have to intervene before the onset of the degeneration occurs in order for gene therapy to work. Now, a number of places are starting to do this, so I've given it a green, even though it probably deserves a yellow. We also need to have a clear understanding between the genotype and the phenotype of the genetic abnormality. So, for example, if someone's born with not maybe an Usher's mutation, but maybe something else that we know is involved in a deafness gene, we need to be absolutely sure that that gene and that mutation is going to lead to hearing loss. Because if we need to intervene before the hearing loss occurs, we need to know that that's actually going to cause hearing loss. But we don't really have a clear understanding in every single case. So that's a yellow, because we have an incomplete understanding. Then we're going to need a custom gene design for every single mutation, even within a single gene. So right now, we've got a few genes that are on the target list, but every single one of those mutations that I listed from the hereditary hearing loss homepage, they're all going to need their own unique form of genetic of gene therapy. And we're going to need a custom vector that's going to get it to its specific cell target, because not every vector targets the same cells. In many cases, as we know from a number of forms of genetic deafness, we're going to have to intervene before degeneration, and that might mean going in intrauterine while the baby is still in the womb. And then lastly, which we don't know, we're going to need lifetime transgene expression, because the underlying genetic deafness may not go away. So this may be where CRISPR-Cas9 ultimately is going to be needed for a lot of these forms of deafness. Next slide. But the amazing thing is that if you go to Boston today, there's two companies, Aquos and Decibel. They're a quick 8-minute Uber ride from each other, as I mapped the other day in Google Maps. And both of these companies have already gone to the FDA for an IND to start gene therapy using otoferlin, that same paper I just described to you a couple slides ago, using the exact same methodology that we described, a dual vector approach. I don't have any IP in that, so again, sadly, another lack of conflict of interest. I wish I had some, but this is really exciting, because we're going to go into patients with our first gene therapy probably within the next year. And based on the results that we've seen in mice, it is really exciting, because I think this has the greatest potential to cure at least this very tiny slice of patients with genetic deafness. But I think this is going to be the first step in a very long line of treatments for a number of forms of genetic disease. Although we have a long way to go, we are just on the cusp of this, but really it's an exciting time to be in this field. Next slide. So gene therapy for hearing loss is coming. It's not coming in 5 years. It's not coming in 10 years. It's literally coming within the next 1 to 2 years. So hold on to your hats. I think you're going to see the first presentations very soon at our next big meeting. Next slide. So with that, I will open this up to questions. I wanted to make sure we had enough time for questions. I'm sure you may have some.
Great. Thank you so much, Dr. Lustig. That was a really great talk. I can start it off here. I have a question of my own, and then I'll keep, people can either speak up or they can write it into the chat box and I can read it aloud as well. But to start off, I was curious currently how you utilize information on genetics of hearing loss, both in your clinical decision making and how you communicate with your patients, given that we're almost there, we're not quite there, that you're offering gene therapy to your patients, but how do you utilize it in your practice?
So great question. So number one, we try to get every kid genetically tested, right? Either we do it here through genetics, or we're starting to do a study where we're working with some outside companies to do that. So I have a frank discussion with the family. Listen, we have no genetic test for hearing loss right now. There's one around the corner for one very specific mutation. If your kid has that mutation, you are golden. You're going to be, he or she will be eligible for a gene therapy study in the not too distant future. For the other families, I say, listen, there's nothing around, you know, and if you're not one of those mutations, you know, we don't, there could be things within the next few years, and you may be a candidate for those as well. So it's critical to sort of to know these things. It's also important for counseling the family, like what's the odds that they may have another child with genetic deafness. So I think that's important as well. The hard part comes when the family comes in, right, and they say, I want to do genetic. I want to wait to get my kid's deaf, right? I want to wait to get a cochlear implant because I want to wait for gene therapy to come down the pike, or I want to wait for hair cell regeneration to come down the pike. That's where the hard conversations come in, because what we know from cochlear implants, that the number one factor that can predict outcomes is length of time between deafness and when you get the implant. The longer you go, the worse the outcome. There is no difference in that paradigm, whether it's gene therapy or a cochlear implant, because it's all about central auditory processing. So if you wait 10 years for gene therapy, you're probably going to have just as crappy results as if you wait 10 years for a cochlear implant. So you still have those critical periods where you have to intervene, beyond which any type of stimulation from the peripheral auditory system, whether it's a molecular approach or a mechanical approach like an implant, is going to have the same adverse outcome. So that's where I think it's important at least to push at least one implant on those patients and say, you want to wait a couple of years, that's fine, but at least get one into your kid.
Thanks. Vivek Kanamori wonders, can you discuss the challenges of delivering enough viral vector to the target cells, especially if you're using multiple vectors that are later combined?
Yeah, there's no question that a single vector is a preferable model, but obviously we have some limitations to that. What we've shown in the lab is you basically have to use super high concentrations of the vector, but that seems to work, right? In the animal, it seems to work great. We don't know if it will work in humans. It does work in primates, non-human primates, and that's as close to us as we can get. So all available evidence in animal models suggests that using a dual vector approach, we can get enough gene into the entirety of the cochlear duct to do what it needs to do, at least for otopharynx. The question happens is, but at the end of the day, we won't know until we go into humans and see what happens.
And I was wondering also, what, in your opinion, are the major outstanding barriers at this time to really making the next step to where this is available in humans? You showed the slide with the cycle and those aspects that are green and those aspects that are kind of ongoing. But what do you think the real bottlenecks are? Like maybe the 1, 2, or 3 bottlenecks that really need to be overcome to make this more widely available and applicable?
Yeah. Well, I do think as soon as we have our first viable genetic therapy for deafness, right, that's going to spark universal genetic testing for everybody with hearing loss, right? So, and I think that in and of itself is going to create a bit of a revolution in our field, because we're then really going to start to be able to do some of the population studies of incidents that we don't have, right? So, for example, otopharynx, right? The best estimates in the papers that have been written, 4 to 8% of cases of non-syndromic deafness. Well, when you try to find those patients, now they're like finding a needle in a haystack. So it's probably 1% or less. So, but it's not until you get every kid tested that you start to really understand what the incidence of these various genetic disorders are. And we might begin to understand some of these poorly understood, maybe there are multiple genes that when they align up in a particular way with certain forms of disease, that's what may lead to deafness. And that may be those are sort of our higher to reach fruit, not the low hanging ones where we've already gotten, where you need multiple genes to lead to deafness. So I think that's one barrier right now, which I think will be rapidly overcome. The second big barrier is just sort of the nature of what we're doing. What we have seen in a number of the mouse models that we've been working on is that gene therapy sort of freezes things where they are, right? It stops further degeneration from hearing. So if you intervene after the hair cells have degenerated, your gene therapy is not going to work, right? Because those cells are already gone. So you've got to really, particularly in the structural proteins, all the Usher's syndromes, those are the ones where degeneration starts really early. And if you don't get every single cell loaded with that vector, you're going to start having cell death early on, and it's going to ruin your results. So I think we have to figure out a way to overcome that, how to get pretty much uniform 100% transfection before degeneration occurs. That's going to be our big challenge in some of our structural proteins. And I think the other big barrier is, if you look at what's the cost to bring a drug to market today, it's about 500 million to 2 billion, depending on where you're looking. Most forms of genetic deafness are what we would consider, rare disease, orphan diseases. It's going to be a hard sell to get a company to sign on for $1 billion if they're going to treat 20 patients with a particular genetic form of deafness. It's not going to happen. So what we really need, we need to reduce those financial barriers of developing and then getting these drugs to people. So the FDA the NIH is going to have to relax some standards. The FDA is going to have to relax some standards, and I think those are going to be the biggest hurdles. That's why we've got to start with the easy ones. If connection's a big one, that's going to be that's a no-brainer, because there's a lot of patients with that. But some of these smaller ones, where maybe easier to treat, is going to be a harder sell.
Anyone can feel free to jump in at any point, but otherwise I was going to ask one final question from me, which is a little bit of a different topic, but related. I think there are a number of residents and fellows on this talk, and you've managed to be both a very successful surgeon and leader of the department, but also really become quite involved in both the basic and the clinical science of this throughout your career. And I think many of us would love to know how you kind of got involved in this type of research and how you've been able to be so successful and continuously involved in it throughout, while also balancing your clinical career.
That's a whole nother talk. So thank you for that compliment. I appreciate that. I think number one, you have to start with a passion for the science. That's important, right? I think all of us in medicine, for the most part, have a passion for the science. So I think most of us are sort of past that bar. Number two, I think you have to put your money where your mouth is. Very early on, you have to kind of get the necessary laboratory experience to understand how to do some of these types of studies. And it doesn't have to be basic science. It doesn't even have to be, it's just whatever it is you want to do, just make sure you put in the time to really understand how to carry out those investigations. If it's clinical outcomes, really, think about a master's degree. You don't need a PhD to do these kinds of research studies, but really assign on to a group of people that really know how to do it and learn from them. So I think that's number one. Number two, for your first job, get the experience early on. So when you get your first job, you can really secure the necessary time in the lab or towards the research that you need, right? You need dedicated time and protected time early on to launch your career. I know it seems hard to believe, but as soon as you show up into the clinic, you're going to get busy. You will not have a problem getting clinically busy. In fact, the biggest problem would be getting too busy too early. It happens to everybody. You want to be the big mucky muck on the panel talking about, how I take out acoustic neuromas or how I take out, sinus polyps. But what's really hard is being the person on the panel talking about how the basic science research is now informing that disease process. That's where you're really going to make a big change. And to do that, you really have to spend the time. You need that dedicated time in the lab. And from the moment your feet hit the lab, you got to start applying for grants, because as soon as you hit that 3-year mark, your chair is going to be saying, I don't know, no funding yet. I'm spending all of this time for you to be in the lab. So make sure you have enough 3 to 5 years of dedicated time. Start applying for grants early and often. Never be discouraged by not getting funding on a grant. Everyone will tell you that you've got to develop thick skin and just keep applying over and over and over again. That's also critical. And number 3, I think the most important thing for an MD is to sign up and make sure you collaborate with a PhD of a big group that has a regular lab meeting and is always there. So when you're not in the lab, your postdoc, your research assistant is constantly working and in a very supportive research environment. You need that strong environment that supports you when you're not there seeing patients. I think that's absolutely critical. And if you look at the most successful people, they have sort of a side project on a bigger group that's doing something different. And I'll give an example. Justin Golub, who just got his first R01, he's one of our junior faculty. We're a relatively small department. We don't have a huge number of NIH-funded labs. We have a few. So he hooked up with the Department of Neurology that's got an enormous group studying cognition and dementia, right? It's just like they have huge grants in that group. So he's studying the relationship between hearing loss and dementia. So he is got the small research project. He got a K award that allowed him to sort of get the necessary time to develop the preliminary data. He's got a huge group that supports him, that helps him along the process. They know how to get big grants. He just now converted that to an R01. He didn't get it the first time, but he got it the second time, and now he's on his way. I think those are the key steps to really making it happen. Getting the good background, being in a good group, being passionate about protecting that research time early on to get the grants, and that's what gets you going.
Thanks so much. Really appreciate it. So I want to respect Dr. Lustig's time and everyone else's. So please, I'll be silent for a minute or so, but then I'll kind of conclude things if there are no other questions. All right. So I think we're finishing up. Thank you so much for taking the time to speak with us today, Dr. Lustig, and it was a great talk. Have a great night.
You enjoy training, everybody. It's the greatest time in your life.
Thanks for doing this, Larry. It was great.
All right. Thanks, Dad.
Great job.
All right. Bye, guys.
Bye, everybody.